So the goal of the research in my lab is to determine the effects of climate change on marine species, in particular changing temperature, declining oxygen, and declining pH in the ocean will influence how animals can obtain and use oxygen. And depending on where and when those conditions change will alter the distribution of species throughout the ocean. In particular, we study how animals are tolerant or intolerant of oxygen, temperature, and carbon dioxide, and how those changing variables with climate are going to influence the distribution of organisms both vertically and latitudinally throughout the ocean. Most of my work is focused on squids, and this is because of all the marine animals, they have among the highest metabolic rates. That is, they use oxygen at the fastest rate, comparable to tunas and the fastest sharks in the ocean, yet they are constrained in terms of how they are able to obtain oxygen. They have a blood that's very sensitive to pH and temperature, and so the way the blood binds oxygen may be limited as oxygen and temperature and carbon dioxide change in the ocean. So we've worked in particular on the jumbo squid, Dipsydicus gigas. This animal reaches very large size and lives in an area that you wouldn't expect such a sensitive animal to live in. It's in the eastern Pacific, where temperature is naturally high at the surface, and oxygen, as you go deeper in the ocean, declines, reaching almost complete anoxia, the absence of oxygen, when you get to about 300 or 400 meters deep. And these animals spend nighttime up near the surface hunting, and then they migrate during the daytime down to about 300 or 400 meters where the oxygen is lowest. And so we've determined that they're able to shut down their metabolism and save energy when they dive down into this low oxygen layer in the eastern Pacific. And we've also determined that their blood is in fact very pH sensitive, but not to such an extent that we expect the increase in carbon dioxide in the ocean, which lowers the pH, to affect them that dramatically. Other projects that we've worked on in the same region are on zooplankton, which are at the sort of base of the food chain just above phytoplankton, providing food sources for the vast majority of organisms on the planet. These zooplankton similarly migrate up and down each day across this temperature and oxygen gradient in the ocean. And we find that they, like the squids, also are able to shut down metabolism when they're in very low oxygen, yet they're very effective at extracting oxygen from the ocean. But we're interested in how their distribution vertically changes as you move from a region with very low oxygen to a region with higher oxygen. And what we're finding is that in the lowest oxygen regions, some species drop out. We don't find them there. Other species are more tolerant and able to extract sufficient oxygen, and that their actual vertical distribution does shift a little bit in some cases. They'll move higher up in the water column, especially at night when they migrate up to the surface. They migrate up shallower than you find them in other regions. So you can actually scuba dive with some of these animals that are more typically deep water if you're in the eastern Pacific where the oxygen is very low. A third project that we're working on has a similar goal, trying to determine the distribution of organisms as climate changes, but we're focusing on 3 commercially important species in the Northeast. In particular, the black sea bass, long-fin squid, and spiny dogfish sharks. These animals are commercially important, and so there's a lot of interest in figuring out how they'll be affected by climate change. So we're looking at total metabolic scope, the difference between their resting metabolic rate and their maximum metabolic rate. So we can put them in chambers and force them to swim against a current, generating a maximum rate of metabolism, oxygen use, and then we can cut that current out and measure the same rate when they're resting. And at a particular temperature, the difference between those 2 rates will be sort of optimal or maximized, and as temperature increases further, that aerobic scope declines. And that scope tells you the maximum amount of energy available for activity, for growth, reproduction, all the things that are important to species. One of the goals of our project is, in addition to determining what the oxygen tolerances of the organisms are, we want to map these oxygen minimum zones. They are currently considered very stable features where the oxygen doesn't vary much over short temporal and latitudinal distributions, but we're finding that there is actually a lot of variability in the oxygen from one cruise track to another. So over very short distances of maybe 20 to 30 kilometers, we're finding large differences in the oxygen available to the animals. And so I think this will change the way we perceive oxygen minimum zones and how we are able then to map the distributions of organisms in relation to temperature and oxygen.